So, as Nathan said, I'm Clint Frese from West Central Illinois. Grew up on a family farm. We did earth moving as well. So I grew up in a dozer, scraper, and as I talk to Nathan a lot, I complain in the last month that we've been dry, and he'll just say, "Be quiet. Don't say that. You don't know what dry is." So, but crops look great here as a whole, and so yeah. So we titled this, kind of advanced soil and sap testing. Using data to find problems and to solve problems. So as I have built products at our company, I've really used a lot of these metrics to validate products, beyond yield. And so how we can kind of put this full circle together of using data, for our business, we're using this internally as well to help kind of design products and to see their performance in the plant and how they react. So. Sweet. So one of the first things I recommend you do when you start this process of using data, we all have limited time, limited resources, and so I believe in, you're going to hear me talk a lot today about buckets, putting your farm into buckets, management buckets. So whether that's irrigation and dry land or manure versus conventional fertilizer, put your farm into buckets, and that's how I start to pull my samples. So for my farm, we've got some manure, we've got some conventional till, we've got some strip till. I will put my farm into buckets, and that's where I'm going to go sample. So whether I'm looking at a biological soil test, a rapid soil test, or a sap, I start with the buckets and I kind of hyper-focus those to see the differences in our management systems. So biological soil testing, the one thing that we have really started on with this is, as a farmer, we don't know what our strengths and weaknesses are from a biological perspective. We see lots of marketing material and magazines online. This product does so many bushels, this or that. As a farmer, I've never really bought into that. I think we've got to look deeper and use data to try to uncover from a biological perspective what is the strength and weaknesses of our soil. And we use Earth Optics and also RiseBio to kind of uncover from a biological perspective how we can do better. What is the strength of our farm currently? What is our weaknesses? Then we can go look for products that align with our weakness and start to validate them out slowly. We have seen great weight. We use a biological program that maybe is going to provide us with a nutrient use efficiency. We can sap our treated and untreated portion and look at the nutrition flow after we implement a biological program to evaluate, hey, what is the flow difference? And then, what we've done internally is look at that across different geographies, across different weather environments. So as the weather changes from a really wet year to a more optimal to a dry year, what is the flow that products will give you to your plant, and how can we uncover what that is? And SAP's been a great validation tool, as well as yield and follow-up test. So the move to plat SAP, and I'm sure you guys have all probably heard us talk about SAP, but I want to run through it quick because I think it lays the foundation for what we're going to talk about today. SAP, what that is, it's the look at the xylem and phloem of your plant. It is actually at the lab, they press out the actual sap or liquid portion of the plant, and is looking at what's flowing or moving through the plant system. Where tissue is a look at, they basically grind your tissue of the leaf, they dry that, and they look at what has happened, or they look at the total nutrients that has been stored in that plant. SAP is a look at what is being consumed. So it's the flow from the root up, and it really tells us, gives us a little different picture that we've felt that's uncovered a lot of things. So yeah, the big thing is they use pressure to disrupt that sap, but not all the cells. So it's really just what's flowing to the plant. The big thing from a probably one of the highlights is it's the mobility of nutrients. So once we get past V6, we want to pull old and new growth in our sap, and that really gives us a look. What is the difference between where is our nutrients coming from to support new growth or reproductive tissue? So in a perfect world, we want our root to support the plant as it's growing, whether that is new growth or reproductive tissue. When we see mobility issues and we see the old leaf much lower than the new leaf, we know that we're already robbing from the old part of the plant that's already been built to support the new part. As a farmer, I think there's big weight and bushels out there for keeping our old growth supported to our new growth until we get to reproduction. When we get to reproduction, we want that plant to start to pull from the old growth. We want that supply to move very, very quickly to support reproductive tissue, whether that's the pod, kernel, etc. Immobile nutrients, these will always show up as the deficiency in the new growth of the plant. These don't move. Once they're parked in the plant, they really don't move. If they do move, there is some data coming out that some of these can move, but it only comes from very critical stress periods in the plant. And it basically means we're under severe stress. So, but these will never move, and we really, when we see deficiencies in new growth, our soil is not supporting that. We need to maybe potentially make an application or think about how we can better our base rate fertility to offset and deliver these nutrients. Nutrient mobility, so nitrogen, phosphorus, potassium, magnesium, chloride, sulfur, and molybdenum, when it is attached to nitrogen, are all mobile in the plant. So these will always show up as deficiencies in the old part of the plant. So again, this is kind of what we do. When we get to V6 in corn, we will pull old and new growth. And so one of the questions I get a lot, especially in corn, when we get to VTR1, where do we pull these? So we like to pull the ear leaf. When we get to VTR1, this picture kind of still shows us pulling the new growth of the plant. We want to get as close to the ear shank as possible to try to detect what that mobility is of the nutrients going in. On the old growth, when we want to pull these, we really want to pull the fully most viable leaf that shows no visual deficiencies. So it's a little bit of a process, but there's a lot that we've been able to uncover with this. So a lot of parameters in SAP. It almost gets overwhelming with as many parameters as we can look at, but we're learning a lot about how these all react. And some of these we look at stress signals. So as we start to look at aluminum or chloride, we do see that some of those are stress signals in the plant, and we're trying to put that all together. In irrigation, those can mean very different things, and I think Jason will uncover some of that. So this is what a report looks like, but at the end of the day, it's about anion to cation balance. So looking at your positively charged nutrients versus your negatively charged nutrients, and the ranking or the relationship between those. So one of the biggest things I do when I look at a SAP report is I look at the negatively charged nutrients and I rank them in my mind. I take chloride, nitrate, phosphorus, and sulfur, and I try to put them in a visual ranking. And Jared Cook that works with this, he has a one-liner that he who has the most wins. So if chloride's dominating the system or nitrate's close to dominating the system, I'm always looking for the weakest link. So what we see a lot of times in irrigation, that's phosphorus being the weak link. And in most cases, it's sulfur on the negatively charged nutrients. As we get into the cations, the positively charged nutrients, the thing I do is I always come down on my report, and I go down here and I move ammonium, positively charged, up to my cations, and I'll do the same thing. I'll rank them. Which one's dominant? Which one's the weakest link there? Cations get a little more wonky. We really don't want sodium being a lot of times it'll be the low man on the totem pole when everything's going right, but that's okay. So I'll move up, and I look a lot on my cation side to the relationship between calcium and potassium. So and on the SAP report, you'll see a potassium to calcium ratio. We've really identified that as a key driver of overall crop yield. So in high yielding corn that gets 270 to 300 bushel, we see that potassium to calcium ratio needing to be close to a 7-to-1 or greater. So This is really explains kind of some of those hidden factors, and we talked about with the nutrient mobility. But again, you can kind of get an idea here where the plants, we've got the old growth and the new growth. And when we have more in the old growth than the new growth, we've got supply within our plant, meaning the root is taking care of everything we need, and we actually have a storehouse of potassium. When we see this deficiency or deficit, that means that root is no longer getting that. So if we're irrigating a lot, especially on this phosphorus, and we know our water's maybe putting problems or threat or outcompeting our phosphorus, we see that as a place where we can come in and really do some good. And I think Jason's going to get into that. So this is probably the biggest aha moment we've had with SAP, is the nitrogen story. So we get 4 big parameters we get to look at. We get ammonium, nitrate, total N, and your nitrogen conversion efficiency. So where we want to be is to utilize our nitrogen that we apply, or that our soil gives us and we mineralize. We want that to go to total N to be able to use that. A lot of times what we see, especially in the Corn Belt, under the logistics that we have, and the cost effectiveness of anhydrous, we see these rapid flows of nitrogen to the plant. So part of that's due to the logistics, I think, but part of it is we have never really put weight on what our soils mineralize year in and year out. How much does our soils give us for free? And the one thing that I think is important that with SAP is and what we're trying to give back, to the group within Calibrated, that changes due to the environment. So when we're really, really wet, we'll see this flow of nitrate and ammonium slow, because nitrogen's mobile in the soil and it's being suppressed down. When we get really, really dry, our soils don't mineralize at the speed. The biological activity slows from the mineralization standpoint, and we see that flow slow. But under ideal growing conditions, this flow is great. And we feel that this has the biggest threat on imbalances in the plant, a big threat on energy burdens to the plant. And with that being said, that kind of pushes us down the line to disease and pest threats. As we get accumulated nitrate, we get really big plant cells, which basically means we just have lots of water in our cells that becomes easy for pest and disease to attack. So this is probably the big one. It's not always the problem, but in ideal conditions, this seems to be a big problem for us. Rapid soil testing, just going to go through this real quick. I consider this a look of what our fields release and the balance of nutrients in our soils under normal rainfall. It's kind of said now that I think a lot of our rainfall has a pH of 6.3 to 6.7. It's close to neutral. There is some mineral composition in rainwater, but a saturated paste test, or the rapid soil test, is deionized water, so water without any minerals in it, no antagonisms, that has a neutral pH. So when you think about this is really what we're after here, is back to he who has the most wins. We want to rank our nutrients, and it really tells the story of competition within our soil system. But the main objectives for me for the rapid soil testing is the ranking, he who has the most wins. So we will divide our positively charged nutrients and our negatively charged nutrients. So the anions, chloride, phos, sulfur, nitrate, we want to look at those and rank them out. And same with the cations. What's really cool about this is under irrigation situations, we can take your same soil test we pull that they're going to run with the paste extraction or the rapid soil, and we can run it against your irrigation, or even your irrigation with like the acid amended fertilizers from Soil Mender, and look for a solution. And it really paints a story of, this is my soil's release under normal rainfall. This is what happens when I turn on my pivot or drip, and this is what happens when I try to amend it. And that, I think, has been tells an awesome story, and I'm sure a lot of you have been doing that. So talking about 3 main dysfunctions today that I've kind of encountered with the data. And the first one that I want to talk about is the soil biological dysfunction. So this is where I started over a decade ago. I started in microbiology, and back probably in 2013, I didn't know what a soil microbe was. So just started making it kind of in my a side shop at one of our farms. and seen some good things, and then seen some things where I didn't have any results as we applied it. So kind of been on a quest to really understand soil biology and its functionality within our soil. So some of the things that I think that have limited us on soil biology over the years is more of a monocropping system where we don't have the diversity anymore. We're more of a corn soybeans rotation in a lot of the corn belt, or it really depends on your area, but it seems to be that is a main factor in limiting the overall diversity in our soil. You know, there's lots of ways to attack this. We see guys that'll plant wheat in a rotation and boom, yields will come up on the following many crops. But I really think that is a carbon biological system that we are activating. So there's lots of solutions from biological side. But the other thing is synthetic fertilizers. They have limited it, not to the point that I think the industry kind of points that seems to be in certain areas of region kind of a war on potash because of its chloride availability. Well, SAP has showed us that manures have way more chloride in them and sodium in them than a lot of our conventional fertilizers, and we'll always see much higher chloride on long-term manure than on guys putting down excessive rates of potash. So there's a lot of fallacies out there that we got to be careful of and watch and validate. And I think that's one of the cool things about all this testing. We can validate this and say, I don't think that's so much true. And that's, you know, as a farmer and somebody that's been developing products, that's kind of been my quest of trying to understand this. But the other big thing that what the data kind of supports is our soil carbon system is being depleted slowly. So I think there's a lot of people out there that want to say that we've got only 40 or 50 crops left before our soils are broken. I don't buy into that. I do think from intensive tillage and intensive nitrogen applications, we are slowly eroding our soil organic carbon, which is the lifeblood of our soil, and keeps that biological system going. So, you know, another way to support your biology is dial in your nitrogen program. You want your biology to work better, get better with your nitrogen program. Over time, that's going to keep our soil carbon at a better level to allow that biology to thrive. But lots of reasons. In the Corn Belt where I live, with our high clay soils, I see heavy rains as one of the biggest detriments to soil biology. We create compaction, we create anaerobic environments, and those really seem to limit a lot of how our biology functions. So lots of different things, but there's a lot of dysfunctions, but it's not everywhere. The better job we do at managing our farm's environment, our physical attribute, our compaction, our oxygen and water in our soil, the better the soil biological system is. So, but there's lots of ways to restore it. Different crop rotations, cover crops, managing compaction, managing drainage is the big one that everybody seems to forget, at least in my area. The better job I do with drainage, the better my soil biology is. So what we do, again, from a biological perspective, I don't know a lot of the farms I get to work on. So a lot of times I'm consulting over the phone, or I haven't necessarily been there. So the first thing we'll do is pull a biological soil test from either Earth Optics, RiseBio, Biomakers, and we'll look for strengths and weaknesses within your system. Then we can start to formulate a plan of attack that's more educated and pointed, and then we'll validate with the sap, yield, etc. But this is kind of an example here of what RiseBio does. So today we can go and pull a soil sample on your farm, and we can run your standard in-furrow program, or maybe a biological system or program that you're running versus ours, and we can start to validate this. So what this looks like, we have on this grower here, he took his control was just starter fertilizer. He then added BioAg E to it. Aminocarb is one of our biological food sources or carbon systems, and then we put the two together with the starter. But essentially what we're able to do is look at this from a scoring perspective, but they give you lots of different metrics from nutrient cycling pathways, disease levels, your mycorrhizal content. There's lots of parameters they look at, but they try to score this out. And this Rise has kind of put a yield to this. So down here at the bottom where you see the response to treatment, the control is a 4.49. We see that BioAg E took it to a 0.03. We actually went backwards just putting the food source on. We went negative 0.02. where we put the BioAg E and carbon together, we came up to a 0.21. What they kind of say at Rise is for every 0.01 to 0.02, that is worth anywhere from 2 to 5 bushels. So this was in western Iowa 2 years ago. This program ended up being about 14 bushel. So it really kind of dialed us into what we should do and taking it to the field. I've seen these where we've got really strong functioning soil where there is no change down here, and that tells me that that farm is already strong on biology, and maybe we're going to look at other things or lower hanging fruit from your farm from a nutrient perspective that we should go after to try to create nutrient use efficiencies, etc. So one of the things I designed these about 7 or 8 years ago. Our process, what we do is we use different compost as a growth medium to house our biology. We extract them and have kind of been perfecting this process and are now patented on our process of making biology. But our goal is diversity in biology. I think a lot of the companies out there on biology, they've really went rifled in their approach, meaning 1 or 2 microorganisms that have one function in your soil. We're trying to do a lot. So we know that there's hundreds of millions to a billion plus soil microorganisms. Our goal is diversity because we don't feel we know enough to be rifled and targeted in our approach. So I want to take you through, this is an RNA report and kind of a summary of BioAg E, kind of the flagship of all our biology, BioAg E breakdown intelligence of our biological lineup. But we look at functionality and biodiversity. Those are the 2 things when I look at trying to perfect our product or continually get it better. We really want biodiversity and extreme functionality within the soil and plant. The first thing we want to look at, how we can get bushels or yield out of our farm or efficiencies, we want to look at the biocontrol pathways. So these are organisms that are native in our soil, that are natural in the environment, that naturally will act as biofungicides, nematicides, insecticides, and bactericides. So we like to look at those rankings in our reports of our product and in our soil. And when we see really weak fungicidal agents in our soil, and I know I've got fungicidal agents in our product, that's an immediate lineup of, hey, this could potentially do us a lot of good. The second one we look at is stress adaptation pathways. So those are organisms that produce different defensive hormones that the plant needs to operate and create its own defensive systems. They also will digest salts, heavy metals. They create biotic glues that protect the root system from different pathogens or heavy metals and salts in the soil. But these are all different pathways that are built in that should be in a healthy soil, but are also in our product. The next one is hormone production. So there's a lot of talk and has been a lot of talk about PGRs within plants and cropping systems. The plant produces all the hormones it needs. In a healthy soil and a healthy plant that's nutritionally fit, it will produce auxins, cytokines, and gibberellic acid to regulate growth. But those are all done in conjunction with microorganisms. The plant through photosynthesis feeds these organisms, and then they go to work producing the hormone, and the plant will take what hormones it needs. Plants are very intelligent at the end of the day in deciding what it needs. The last one, the one that probably has been talked about the most in the biological space, is nutrient pathways. So these are organisms that will take inorganic or unavailable nutrients and then convert them to available nutrients and transport and assimilate them to the plant. But we try to really focus on the carbon cycling within the nutrient side, as well as all 15 nutrient pathways. So the big benefits to BioAg E that's kind of behind the scenes, it's very balanced bacteria to fungal, big protozoa load that act as predators against bacteria, and we've got extreme functionality to do a lot of different things. Breakdown, all our biological lineup is extremely diverse. What we've done with breakdown is essentially just add more organisms that have cellulose and lignin degraders. So all those biocontrol pathways, the nutrient pathways, the hormonal pathways are all still there. We just kind of put these guys on top. But again, you take my area where we got drainage under control. A lot of my areas, still disc ripped a lot of disc ripping. We are oxidizing the soil through the disc ripping, where we are creating an environment where these organisms thrive out of the gate. Not a best fit for BioAg E breakdown. We go into Nebraska, where there's a lot of areas where we're finding 2- and 3-year-old corn root balls and stalks, where we're in more of a no-till, strip-till environment. This has been a big play that has added bushels. And one of the things that we've learned the most from the vegetable or table crop industry, to break disease cycles, all our diseases harbor in our residues. So if you're seeing fields with extreme disease problems, this fall pass can be one of the biggest, most important passes you can do to break that cycle of disease. So, and then the last rung that we've created, and probably our fastest growing, is the intelligence seed treatment lineup. So it's everything I've described about our biological lineup into a more concentrated condensed seed treatment. This is our fastest growing lineup. It's the most cost effective to apply. We've got plots all over the US with this, and have been really happy with what we've seen. So a lot of what we'll see with intelligence, we will still see this nutrient cycling, and we can pick it up in the soil test the fall after. So seeing great yield gains. We can protect that rhizosphere or that plant and that seedling early in the season, but we seem to do more longer. Most seed treatments have a 18 to 21 day window of protection. We feel we get that plus long-term nutrient cycling within our system. So, but this is kind of how we've validated. We've spent a lot of time evaluating nutrient use efficiencies within biology. We see big differences in soil tests as we apply. So what you're looking at here is a 3-year study where we've reduced fertility, P and K fertility, by 50%, 45 to 50%, and we basically see no soil test change. And so that's the big thing we've validated. There's a lot of companies that promote, hey, use our biological, you can reduce fertility by 50, 100%. I'm not in that camp. I think that when you have to validate from a standpoint of your environment, your geography, your soil, and find your nutrient efficiency curve. We've got guys that haven't applied fertility in a decade. Their soil tests have not dropped. I've got guys that have only went 2 years at a reduced rate, and their soil test levels start to come down. So understanding how good our soil is and the bank that we have in our soil is important when you start to go down the road of nutrient use efficiency. I like to show this slide. I think a lot of farmers do get held up, myself included, on soil test levels, trying to get to a certain level and hold it there. I always say, give me 12 months to pull your soil test, and I'll make it look however you want, high, medium, or low. So this is essentially just a chart showing that in season, when our crop is actually growing and we're actually mineralizing and our biological system is moving, our levels are usually 40 to 100% greater than a fall baseline level of fertility. The environment matters. I like this slide going back to as you incorporate a biological system and you focus on that we can reduce fertility by X. So a lot of claims on that. What we feel is, you look at this is an average of our plot. So this is control, would be a starter program plus starter plus our BioAg E. So in 2024, we had a really slow start to the spring, wetter spring, where this plot was. And total in where we applied the biology was increased by 18%. Potassium was 15%. Phosphorus 25%. 2025, we had excellent soil to oxygen ratio. We had warm soil conditions. We applied the biology in furrow. We had a 48% increase in total in that plant sap. We had potassium 40% higher than the control. Phosphorus 60% higher than the control. So the environment matters. So when you see in a farm journal, you see an ad that says, hey, we can reduce fertility by 50%. Question that, because the environment dictates how strong our microbiology works and how fast and robust it works, and it's going to dictate the flow of nutrition. Some of our plots, I'm going to kind of speed through this. We can talk that, but we do see pretty consistent yield gains. This is a field that we went to several years ago. We applied a biological program, seen 0 bushels. Went out that fall, kind of evaluated it. The field had extreme drainage issues, extreme compaction. So we came in, we corrected the compaction, put in a whole bunch of drainage ditches, came back the next year. We changed the environment of that soil, then applied the beneficial inoculum to get that to go from anaerobic to more of an aerobic system. And we've seen really big yield gains. We had almost a 20-some bushel gain here. But until we fixed the environment, there was no gain to be had. The system, the biological inoculum could not shift the system on its own. So the environment, I know you guys will probably keep hearing me go back and back to the environment. The environment is critical. If you can address the environmental conditions of the field first, a lot of this stuff and your biology naturally will just take off and work for you. One of the big things we teach and preach on is the Gause's law, or the law of competitive exclusion. This has been known since the 20s, and it essentially says when you have two species that compete for the same resource, the one that the numbers that get at the highest level wins. So if you have Trichoderma hazardum, a beneficial species, and you have Pythium, root rot, once you can get Trichoderma hazardum over the total CFU or load, disease load of Pythium, it's going to fall out a bed. So that is how our fields work. And back to the environment, when we have a field that is compacted, is too wet, doesn't drain, you are going to allow that pathogen to thrive. So this is essential. What I think is going to be a lot of the future is talked about. There's no chance of resistance to getting our soil to function properly and outcompete pathogens. So one of the things I've noticed in table crops, nuts, fruits, nuts, and vegetables, a lot of the times we've overused fungicides to the point where we're losing efficacy of those fungicides. And a lot of the nut growers we work with are coming back to these principles of plant nutrition and soil biology to work with their disease problems. So, but this is an example of Fusarium and how BioAg E, treated and untreated, can really lower that pathogen threat. This is how we use, this is a report from Earth Optics, and this is how a 2,000 acre study we did on a local grower, and we essentially looked at 2,000 acres treated with BioAg E for 2 or 3 years and untreated, and then we looked at the disease profile of that farm. And we were able to pick up some pretty big differences. So Earth Optics does a really good job of color coding this and making it easy. When you see a red bar graph, that is a threat to the plant that has the potential to limit yield, that disease. When you see it in that yellow or orangish color, that could go either way. We could have a threat to that plant, depending on if the environment shifts, or we could not have a threat. When you see these light green bars, no threat to the plant. Those disease levels are very, very low to not really have any issues. But so when we look at products to evaluate, or we look at system changes within our farming system or management practices, we have ways now to evaluate our disease threats. And we've used this on some bigger operators, guys that were using 2 and 3 shots of fungicide. We've been able to wean these guys down to maybe just a late season pass from a disease protection standpoint. This again on soybeans, and we don't do every disease well. There's some here with sudden death, Phytophthora, some of those we don't do well, but as a whole we seem to really do well in rooting diseases. I've kind of hit on this earlier, breaking the disease cycle with biology. And when you really think about where biology, your disease ends up, as our plants weaken and they start to senesce later in the season, disease almost always comes in. And it harbors in this residue. And we feel this is an excellent inflection point to go in and start that out competition in the fall. So a lot of guys think, we got to, with synthetics, it's very fast. We use a fungicide, we go apply that. It's quick. Its ability to stop a disease is very, very quick. Biology is a different mode of action. It takes time. So pre-season, pre-plant, I think is an excellent time to kind of go and start to think about some of these breaking disease cycles. So this is probably a really cool, this is Laurel. This is with Taylor Seeds we work with. One of their main concerns when they first reach out, high yielding corn area, but they had 2 and 3 years worth of residue in the field, and they was having a lot of rooting disease problems, a lot of uneven emergence, even with planter attachments and row cleaners. And we started with the breakdown product, and this is a perfect example of taking the problem and putting the solution with it. I got areas we got full tillage systems. This is not necessarily the fit, but in their geography and strip till, this was a very, very good fit that has been very successful. So locally, we had in conjunction with our local university and some interns within one of our dealers, we did a bunch of these residue waste strips, but essentially they collected like 3 by 3 squares, collected the residue. We kind of come up with a 1.37 ton reduction in residue from the application of breakdown, and then we've seen a pretty good yield bump from that. I like to point out that we have these hog buildings back here. Because of the excessive nitrogen, we do get excessive plant growth, so it's another good fit for breakdown programs. This kind of intelligent seed treatment data. This is from North Dakota. Seen 9-10 bushel on Durham wheat. Seem to do really well with intelligence lineup on small grains. The one thing I want to show where we're at today, and some do's and don'ts with biology. So the one big do, in most cases, we like a carbon or food source applied with the biology. Seems to add some serious synergy. In high fertility manure situations, you want to stick with just a carbon source, so no stimulation. So if you see anything with amino acids, those type of things in products, we want to steer clear of those in a support package with biology. What we have really done and pushed a lot this year, and we've worked with NutraDrip on this, is their effluent manure, and putting that with BioAg E and Backbone. We've got a lot of internal plots where we're playing with this in a big way, doing lots of different studies from aeration to different time frames of putting the BioAg E in the manure pit, putting in an at application or before application. We're looking at dairy versus swine, different micro packs to go in. So going to have a lot of data come fall from a lot of different states to see how this works. But the big thing is there's a huge cost savings over a lot of the nutrient or Helena starter programs that we go up against a lot. And that's what this is. This is in southern Illinois, southern Indiana. And we're essentially looking at, I believe, a starter with zinc program versus just the effluent manure with a little bit of backbone and BioAg E. Backbone's our carbon product. BioAg E is, of course, the biological. But some really big differences. I think when the dealer got to this, we thought that there was a different planting date here, but it's just the difference. Very low carbon soils down there. So we're talking 1%, 1.5% organic matter. This farm had never had manure history. We feel that that's going to be a very big fit for this. And one of the things I think NutraDrip has discovered is the effluent manure, while it's really low in nutrient composition, they're all complexed with carbon. So the availability of that nutrition is very good, and it's very common dominant. So when you run 3 or 4 gallons, it's running like it's like running a big shot of humic and fulvic. So some pretty cool stuff, I think, that shows some pretty good promise. So the next thing I want to talk about is herbicide stress limiting crop yield. And I'm going to say I think it is a lot, but I don't think it's every time. So the better job on the better ground we have, I think our crops under ideal conditions handle herbicide quite well. But like for my area, we go from heavy clay soils or prairie soils to very highly eroded timber soils, clay hillsides, etc. And we'll see plants seem to stall out for sometimes 2 to 3 weeks, especially when we get hot, dry weather or we're excessively wet and cloudy. When the environment isn't perfect, we really feel that this is a pain point that we get asked about, hey, what can I do to ride along with my herbicide application? So when a plant has to metabolize herbicide, the first thing it has to be done, it has an oxygen atom or molecule has to be added to that synthetic herbicide. From there, it has to be as a conjugation phase, where essentially amino acids or carbohydrates are added to that synthetic herbicide that is being inside that corn or soybean or that plant, and then it's sequestered in the cell. So it's quite the process of the plant having to take that herbicide and metabolize it. And when we get environmental conditions, this process gets very, very slow. So one of the things we did going back 5-6 years ago was our first year really sap testing. And we went out and we pulled saps ahead of our herbicide application, and then we pulled them 3 days later, 7 days later, and we looked at what happened when you spray, say, Callisto and Dual and Atrazine on a corn plant, or you spray Liberty, Roundup, and List on a soybean plant. What happens nutritionally to that plant? And what we came up with was, as a whole, calcium, sulfur, potassium, and overall carbohydrate load just drops in a big, big way. And we really think that is the plant's way of metabolizing that. It's that way of sequestering that herbicide. So we went to work developing something. ATP Pump is a product. It is the main function is it's derived from corn. So it's derived from distiller's grain, and then we've added several other things to it. And it is a biostimulant, so it's an amino acid grain extract per se. But the one thing that we did get lucky on is ATP Pump increases potassium, sulfur, calcium, and overall Brix levels. And it seems to really metabolize, does a really good job at metabolizing herbicide well. We've seen a lot of visuals over the last 3 or 4 years where we see a lot greener color. This has been a 1 to 2 quart use rate product. So last year we worked with Foresight Agronomy, Dr. Elaine Ellen Harris, and we did gene expression work. We wanted to see, when you spray a herbicide, what happens from a gene standpoint. When we add ATP Pump to it, what happens from a gene expression standpoint. What genes are turned on, what genes are turned off within the plant. The big thing we learned about ATP Pump is its ability to, as a photosynthetic activator. So it increases plant nutrition and plant metabolism, and it increases photosynthetic pathways. That is that mechanism to sequester that herbicide quicker. The other fit for this product, once we kind of did that, is in cloudy, cool conditions, wet conditions. If you've got fields suffering from overall wet conditions, you see yellowing happen. We're at that point in time. We really feel that I've seen a lot of great 72-hour green ups later in the season and some big yield gains from it. But the cool thing about doing some of this work, we realized that 1 pint was the same as spraying water, she said. She said, I might as well just have sprayed water. The other thing, from a gene expression standpoint, a quart is pretty strong. 2 quarts the most effective and will last up to 28 days in the plant of activating those pathways. So, but some of the stuff we're kind of doing behind the background. The last thing I want to talk about, and probably the one that we've talked about the most forward-facing, is problems utilizing nitrogen. And it's something that I think without SAP, we probably never would have really found or uncovered, and probably one of the biggest yield limiting factors that we've been able to address, and potentially saving points for the farmer. So looking at, this has made us back all the way up to look at mineralization rates of our soil from a nitrogen standpoint, as well as reevaluating our pounds to the ground of nitrogen. And I might add this from, as I've worked with growers in Georgia, I can't reduce my N on 0.8 organic matter soils the way I can under a 1.5 or 2%, or what I can do in Minnesota under a 4 or 5% organic matter soil. So it is good to go through this exercise on your own farm, find your own kind of inflection point of where we can get to. But this, back to nitrogen and the disease pest triangle. So essentially what happens is when our plant can't convert nitrogen in the root system to a protein, it is stored inside the cells of our above ground tissue. And when that is stored in our cells, that nitrate and ammonium trigger a hormone called Gib acid. Gib acid controls cell elongation. So when you go out and apply manure on a field for the first time and you see that crop height response versus maybe a conventional fertilizer system, that is that nitrogen triggering Gib acid, creating that cell elongation. When you have a cell elongation, it's like a balloon. Jared explains this as a balloon. Essentially what that means is there's more water space within our cell. So when we have a disease spore that lands on our tissue, and it's trying, what a disease is trying to do is go systemic. It's looking for water. And that will puncture that cell and go systemic. The tighter our cells are in our plant, those disease spores cannot find water. Insects do not like to feed on plants that aren't water rich. So when we talk about nitrogen has the greatest effect on the disease triangle, that's essentially what we're talking about, is that excessive growth and unutilization. Big energy demand. I think a lot of the universities and data point that there's anywhere from 11 to 15% energy demand to convert this unprocessed nitrogen in the plant. But essentially what we've found over the last few years is it takes carbon, carbohydrates, sulfur, copper, iron, zinc, and moly to convert a lot of these. Phosphate or phosphorus is on there because that is the energy of the whole system. We break that one and all the other ones don't matter. What I have found for most of the Corn Belt, and especially what I get to see under irrigation, to me this is the most important one, phosphate. You got to keep it first. So if we're trying to utilize and focus on nitrogen conversion, we have to have phosphate in there first. Once I get this correct in a lot of my dry land applications, I will always go to sulfur too. If sulfur is not right, you can't just go out and apply 4 ounces of moly and think that we're going to get to a place where we want to be from a nitrogen conversion efficiency standpoint. And the carbohydrates are important. Essential carbon is critical to this whole pathway within nitrogen assimilation. And so this is how we evaluate this. We go back to the sap. And from a quick standpoint, we can look at this nitrogen conversion efficiency. We've preached, we really like that in a 90 to 95% conversion. In corn, if I can keep this above 82 to 85, I'm pretty happy as a whole, especially through our rapid uptake period. So early in the season, when you plant from emergence to V6, if we can keep this above an 80-85%, I'm very, very happy, because when soils initially wake up, we are going to get a rapid flow from mineralization plus what we've probably applied pre-plant. But essentially we want to keep especially ammonium, in my opinion, under 50 early and especially early R stages and reproductive. And nitrate we would like to keep sub 150. It gets a little tougher. But really both of them, if we can keep around 50, is a good deal. So, but when you see this problem happen, going back to the environment, it gets dry, I'll see nitrogen conversion efficiency get really good. It gets really, really wet, I'll see nitrogen conversion efficiency get really, really good. But when we get wet, I'll see total N get really, really low, because we probably pushed nitrogen out of our soil system and we're getting to a low point. And we probably need to apply more nitrogen. So when we're ideal, we have good oxygen soil levels, this seems to be the problem, I would say, over half the time, 50-60% of the time. So part of our solution is the essential carbon. So Backbone is a diverse fulvic product. There's 4 different fulvics in there. From all different types of mines. So we're after the diversity there, and then there's a lot of different simple organic acids. So essentially what Backbone does is create double bonded carbons, and those have the highest energy sequence to help put the essential carbons for this process. The next one we've created is this N-Plex. So these are all sulfate based micros, and that's how we're getting our sulfur out of this product. And then it's copper, iron, moly, and zinc. And so a lot of times early in the season when we see nitrogen conversion efficiency get below 80, below 75, if we run a quart of this and a quart of Backbone together, we will increase nitrogen conversion 20 to 30%. And that will hold for about 12 to 14 days in our SAP analysis. We'll do this again in reproductive if it's still a problem, and it's kind of been our go-to recipe. What we've seen is we've had a lot of guys this year just run N-Plex. They've just run the carbon or just run the micronutrients that are essential in the conversion. We only see a 6 to 7% conversion efficiency. It seems to take the carbon to move it. So as we've evaluated 40, 50 sites, I think what we'll probably have next year is Backbone and N-Plex put together into one product, because it does not seem to work and drive the conversion efficiency without both. Lastly, I think what SAP does is allows us to dial in our in-season nutrition program. Most of the growers we're getting to work with now and farmers we're getting to be on, we're leaving dollars for in-season nutrition. And I think that if you look at 10 years and you leave a budget for in-season in dryland acres, 7 of those 10 years it's going to pay you back. There's going to be 3 of those 7 where you could have probably front-loaded it all and it wouldn't have made a difference. Under irrigation, I think this gets more and more important to reallocate dollars from your fall budget to go in almost all in season, or very little in the base rate, enough to get that plant off to the races. But differences between dry land and irrigation, but dialing in crop nutrition has been big. So a lot of times when we side dress, we just want to throw nitrogen and sulfur. In this case, potassium was worth a lot of bushels. This is the detent from Y drops at the end row, and we kind of had pictures of this all the way along a field where we had an 8 or 10 foot kick-on lag. But this is a perfect window. You know, this only lasted about 2 weeks where we've seen this awesome plant health difference. But on a dry year, getting potassium in the side dress was worth some pretty big things. And I think this is where SAP, going back to the ranking, he who has the most wins following this law of the minimum can really do some good things. You know, this was 3 years ago. We was extremely dry in this situation here at application. Rain took back off. Potassium was the loss leader. Nitrogen was not. You go fast forward to this year, and this guy's area, nitrogen's the loss leader. He had 20 inches in June. So knowing how the environment affects nutrition is important, and I think that's where some of these testing metrics can really dial us in. So we do a lot of things at Calibrated, but the one thing we really try to focus on is our research. We're all a group of farmers trying to learn and design better systems and better diagnostics to help us all learn, and sharing that back. We do a lot of different custom fertilizer blends, and we're focused on driving yield, profitability, and working now a lot on nutrient density, focusing on different forages and trying to increase our forage content to eliminate different rations we can put into our cattle from dairies, and then also working with a lot of vegetable growers on trying to increase nutrient density through better management of biology and nutrition. So with that being said, if there's any questions, again, thanks for having me, guys, and for letting me talk this morning. So, oh, why would you see a pop from an application of a gibberellic acid? Yeah. That's a good question. So years ago, I think a lot of the PGR, especially on the growth hormone side, so the cytokine, auxin, and gibberellic acid, stemmed from a gal that was hired on with Stoller, USA, and they really dived into the perfect hormone ratios within cropping, especially on the grain side, so and vegetable side. But what they came up with was the ideal ratio for corn was 2 parts cytokine, 1 part auxin, 1 part gib. And the industry as a whole has followed that from a growth hormones perspective. I'm trying to think of some of the other ones on the market. Jason, would be Ascend, and
yeah, that's all. They're all Stoller knockoffs.
They're all Stoller knockoffs. Yeah. So that is the ideal ratio. And how I try to describe this is if you're trying to grow forage, you want gib to be triggered and you want that growth. And so a lot of times when we are managing forages, we want some nitrate in the system to trigger that gib acid naturally in the plant to create cell elongation or biomass. When we're trying to grow reproductive tissue, that is when we want to focus on cell division. So the cytokine hormone, to me, is the most limiting growth hormone there is today. So cytokines are primarily produced in the root. And think about sugar allocation or carbohydrate allocation from a plant. When the plant first starts, the root takes top priority for sugars. As soon as that shoot gets above ground, the above ground portion takes the hierarchy for sugar first and foremost, with the root taking second fiddle. So when we have a cool, cloudy day and the plant isn't photosynthesizing and creating a lot of sugars, the above ground tissue is going to get all of the sugars. Cytokines aren't going to be produced. It's going to produce your auxin hormones are produced above ground. So having that ratio is important. But I think from a hormone perspective, they can, what I'm seeing is hormones will help nutrition get into the plant. They will speed up in the rate in which they use that nutrition. But I think it's on high yielding crops. I think there's much more low hanging fruit, whether it's evaluating your irrigation water, dialing in nutrition. So a lot of our high yield growers will use hormones. We'll use hormones, especially like where we're cutting hay all the time. I'll use hormones.
Like when the plant's locked up, like in the sodic soil, you can get a nice pop from that.
Yeah. I think there's some.
Yeah, I go back to with hormones. You want the plant naturally.
Yeah.
That's more of a cytokine lockup, or to the root.
Yes. So whether it's boron toxicity or sodic soils, it wants to limit cytokine because it's a root dysfunction that's stopping that production of those cytokines. So addressing that sodic, if we can address and complex that sodium or boron toxicity, whatever it is, that's the ideal thing, to let the microbiology and root proliferate those hormones on their own. But what we've seen, just mixed results with PGRs as a whole. I mean, I've seen big yield gains from it, and then run the same program the next year and nothing. I think it's a timing issue that I don't think we have the diagnostics to understand the exact timing of it. I think timing's probably pretty critical.
It is, but it goes back to the root causes. Why aren't we producing it in the first place?
Yeah.
Much to that point, with the root development and with the acidic soil. But what's your point with nitrate? I mean, you could be throwing if you're having a hard time with cytokinin already, and we go dump 30 gallons of 32 through the pivot, but we just set that even further. So I think understanding the root cause of that is more the issue. So
it's complex. What you're talking about is very, very real. But a hormone's going to last, 7 to 12 days in the plant. So an artificial one that you're going to apply. So one of the big negatives, I will say, the industry as a whole, and kind of leads into your question, but everybody's really promoting amino acids and biostimulants, and you'll see all these kelps and different plant extracts. The one thing I want to say is we are basically taking all these biostimulants in the background and hormone profiling them, running an amino acid profile on them. And what you're finding is a lot of these kelps that they promote forward-facing the amino acid package and how beneficial that is for the plant is loaded in phytohormones. So there can be high levels of auxin, cytokine, and gib acid, but for the most part, all these biostimulants are loaded in salicylic acid, sometimes jasmonic or different brassinosteroids. And essentially those are the defensive hormones that trigger what they call ISR or SAR. So either that's systematic acquired resistance of the plant. So that's the plant's natural ability or its defensive system. But just say your plant's in growth mode and you're going to go apply a kelp that you don't know is jacked with salicylic acid in the background. You can trigger your plant to go from growth to defense and take energy away from growth and actually create yield to go backwards. So I probably had 30 phone calls last fall and winter from biostimulants taking yield backwards instead of upwards. So the one thing I think is important to educate yourself on is there's a lot of stuff hidden in these biostimulants, and they're all byproducts of something that's very, very cheap, that from a 1,000-foot view look very effective to the plant, like they're all things the plant needs. But we've kind of backed up and just went, hey, let's get our biology right, let's get our carbon and nutrition right, and the rest will fall into place. And so is there going to be times where we can use some of these products under extreme stress events? Absolutely. But I think trying to get to the root cause, as Jason says, is where it's at. So, yep. Good question.
What's your opinion on like long-term no-till maybe needing to come in with some sort of like disc ripping or something like that at some point to, you know, loosen up these soils and stuff? I feel like we're kind of, I'm up in southeast Nebraska and we've been no-till since about 91 or so for the most part. I mean, very little tillage. If we do, it's to fix ditches or whatever. Yeah. I'm almost kind of feeling like we're kind of at the point where maybe we need to go in and do some disc ripping or something like that, loosen that soil back up to kind of help jumpstart some of this stuff, break up some of that compaction, and then do something like that.
I'm a big proponent of that. I mean, you know, when we, you know, I'll give you an example on my own farm. We did min-till, strip-till for a long time, tried to do no-till as much as we could, but I would sometimes push stuff with a VT or strip-till bar, wait so long to plant, and then it was getting late May, and we would push stuff, and we would create compactions, and then in our rain with our clay soil, or our environment with our clay soils, rain compacts really a lot for us. So I see stuff pop big time on breaking that. A lot of our vegetable network we work with, a lot of them will even plow every 10th or 12th year. I mean, they're using cover crops, they're trying to do strip till, min till as much as they can, but they'll still reset when it's needed. So I don't necessarily think it has to be every farm, but I do think it's a big deal, a really big deal to reset it.
And then what can be done about chloride levels? I mean, every test that we get is extremely high chloride.
Yep.
I mean, is there anything at all that we can do to help manage that?
It's just overall stuff you're already doing. I mean, so the long story short, we need more carbon in the system. Is the true long-term fix. So whether that's reducing tillage to not break that carbon, you know, as many times. We feel the more we shift, a lot of our data is pointing the more we shift to a biological system. So that does not mean we're ignoring nutrition. You know, nutrition is at the forefront. We have to grow healthy crops to get more carbon in the soil. So, but as we shift to a biological system, I think applying that active carbon later in the season, and so we're trying to find more cheaper, cost-effective measures to apply more fulvic acid. I think that is part of the key to keeping that biological system strong. One of the things that doesn't get talked about a lot, but as you move to more of a biological system and you dial in nitrogen rates, the one potential threat you have as your biology gets really active, the biomicrobes can turn on your plant as a carbon source late in the season and cannibalize it. So I think carbon late season is part of the piece. If you can make it work, cover crops, I think getting more carbon deeper in your soil and just building that pool is where it's at. But to me, you know, we got to also look at the metrics we have. You know, I love what New Age has done for us in SAP, but how factual is the chloride level? We know there's a lot of chloride in our plants, but what level is it really, really toxic? Or is it a stress indicator? I know in irrigation that differs, you know, because you got to look at what your water is and what its composition is. But in dry land, I'm starting to look more of chloride as a stress trigger. I see it get way too wet. I mean, when it got wet from, you know, eastern Iowa all the way through Ohio in June was 12 to 21 inches, 25 inches in June. Chloride went through the roof. And I really think that's a way of, you know, some of the textbooks and articles we've read, that is a way of the plants trying to make osmotic balance. Not a full lot of science there, but that's way is trying to limit water uptake. So that every situation is different. Irrigation water, is going to be different, than what I'm trying to describe. So I don't have a good handle on the chloride in the sap, on what level is really, really harsh, but it is no matter where you're at, it seems to be a parent material of our soil in the U.S. Chloride is a dominant ion for sure. So.
Very good. Thank
Yep.
you, Clint.
Thank you.
Something that I find fascinating too is like, as farmers, right, we all get bombarded with lots of different theories and products, and then we sit here and we apply things and we're like, it worked or it didn't work, and trying to understand and reveal, explain some of the why. And I think with some of the things Clint talked about here, with some of the basically the soil DNA testing, rapid soil plant sap, that helps us understand the environment that we're in more than just it was wet or it was dry, and helps us to actually measure what's going on in the plant. You talked about doing some of those sap analysis before herbicide and after herbicide, and then some of the things that we're now starting to bring alongside in the tank with the herbicide mixes of being able to see that actual effect on the plant, which helps us explain then what's happening when you look at the field monitor. So I think that's a fantastic tool, and appreciate your time